Alrighty, today I'm going to talk to you about Unit 3, biological macromolecules, and Unit 4, the basis of the cell. Right now I'm going to go over Unit 3 summary with you. Alright, so the first thing that you need to know is biological macromolecules are all carbon-based. They all contain carbon to hydrogen bonds. Um, the reason why carbon is so important is its unique atomic structure. So as you know, atomic numbers also indicate the number of electrons each atom has. Carbon's atomic number would be 6. So that leaves 2 electrons on the inner electron shell and then 4 on the outer. This makes carbon an ideal candidate to bond up to 4 times. So as this illustration shows, there are only 4 electrons in the outer ring, allowing it to bond up to 4 times. In doing this, carbon has the ability to form chains, branch chains, and then also even rings. And as we know in biology, structure really determines function, and this makes carbon a pretty diverse atom. Next, I'm going to talk about the 4 macromolecules that we're going to go over. These are the only ones present. I don't know why I made it sound like there's more. Anyways, first one being carbohydrates. This is really primarily for energy storage as well as cellular communication, the monomer of which is going to be sugar. Monomers are the small blocks that when assembled make these larger macromolecules. The next would be nucleic acids. These are information storage. They're found in the nucleus of the cells. They're really like the blueprint, so to speak, of a lot of the cellular functions, and therefore they also are kind of considered the control centers of the cells. Next, we have lipids. These are for energy storage as well as the boundary. They compose they're the primary composition of the cell membrane. So the phospholipid bilayer, that is lipid. Their monomer is going to be fatty acids. And then next, of course, we have proteins. So these carry out essential functions of life. They can be enzymes, so they speed reactions. They can assist in breaking down things in order to speed up reactions. They can be signal carriers, a whole variety of things really. And their monomer is really going to be the amino acid. Okay, so real quick, I wanted to touch on functional groups. The first being nonpolar. So what does that mean? That means, of course, they're hydrophobic, being that they don't carry a charge. And they are also hydrogen-carbon bonded molecules. So 2 examples of such would be methyl, which we have here, and then phenyl, which is over here. Next functional group that you need to know about is going to be the polar functional group. These, of course, contain a charge of some sort due to the electronegative atoms contained within the molecules themselves. So these contain nitrogen, sulfur, or oxygen. This is also going to be, let me show you what I have here. So alcohol, these are going to be in amino acids and carbohydrates. We have thiol, this is part of protein. We have carbonyl, which is part of carbohydrates. We have ester, which is going to be found in lipids. And then last but not least, we have amide, which are also found in amino acids. And then the last functional group is going to be the charged functional group. These are actually acids, so they form ions by releasing a hydrogen, a positively charged hydrogen ion. These are primarily going to be seen in amino acids. Actually, the first one is actually a amino charged functional group there. We also have carboxyl, which both of these are found in amino acids. And then last but not least, we have phosphate, which is very important in like I mentioned, that phospholipid layer. Okay, next we're going to dig a little bit deeper into these individual macromolecules. So the first we're going to talk about is carbohydrates. They have a ratio of 1 to 1, meaning they have carbon, one carbon to 2 hydrogen to 1 oxygen. So they tend to look like, so 1 to 1. Here's an example of a carbohydrate. This is a simple sugar. These then look like this. They have these carbon chains with the hydroxyl functional group. That's what this is. And then a hydrogen branching off of it. Important to know, so the monomer, like I mentioned, is going to be sugar. So these monosaccharides somehow need to get together in order to make disaccharides and then also polysaccharides, so the larger sugars, so simple or complex sugars. In making a complex sugar, all these monosaccharides are going to bond together somehow. So how is that going to look like? It is through a process called dehydration, right here. So these are two sugar molecules, so 2 monomers. If you extract H2O, you then get a disaccharide. You do this multiple times and you're going to end up with a monosaccharide here, as illustrated above. Likewise, these molecules can also do the reverse effect, so they can go through a hydrolysis. So in adding the water, these would then break apart. Next one we're going to talk about are going to be lipids. So lipids, you really need to know that they are hydrophobic. You may have observed this if you ever tried to mix your olive oil and vinegar salad dressing. They just won't do it. It's because the oils are hydrophobic. The first one we're going to talk about are triglycerides. What that means, obviously tri means 3, and glyceride stands for glycerol. It's part of the functional group we just went over. Sorry, I just had a moment there. Anyways, so and then the 3 stands for 3 fatty acid chains. Let me show you what that's going to look like. Oh, here it is. All right, so lipids. Here's a triglyceride right up here. We have the glycerol head and 3 fatty acid chains. Important to note, so some of these are squiggly, some of these are straight. The straight ones would be what we would call saturated. The reason why they take on this shape is they are a carbon chain with every single carbon bonded to a maximum of hydrogen atoms. So it would be saturated with hydrogen. These kind of kinked tails here would be what we'd call unsaturated. So what is happening here is the carbon is forming double bonds with other carbons, lending a kinked nature to the tails. That is why unsaturated fats are traditionally more healthy for you, is because the kinked nature allows them to stay fluid, not to stack easily, and then also to be digested and broken apart a little bit easier as well. Lipids also contain steroids, hormones, and phospholipids. So I drew a little picture here of a phospholipid. It also has a phosphate head and then 2 fatty acid chain tails. These are very vital in the cell membrane. So the phospholipid bilayer, there's actually 2 layers. The hydrophobic tails are forced inside and the hydrophilic heads are on the outside, and it's almost like a mosaic, so it's fluid. They're able to move around, but they're so densely packed due to the hydrophobic effect that really only certain things are able to permeate it, such as small gases, small nonpolar molecules, etc. So that's kind of important to know. These fatty acid chain tails are attached to the glycerol head, or the the glycerol head, by hydroxyl functional groups also. So keep that in mind. Let's see. Now we're going to talk about proteins. So proteins have a lot going on with them. First off, the amino acid is the monomer. The amine group, well, I didn't really draw it on here. So like we showed before, we've got the amino and then the carboxyl. So what this looks like is, this right here would be an amino acid. We have amino, carbon, and then a carboxyl acid down here at the end. And this right here is one amino acid. These amino acids are then covalently bonded through peptide bonds. This occurs through dehydration once again. And also important to know, we've got some different structures going on here. I've labeled that is representative of a primary structure. It's the amino acid sequence itself. Next we have here
kind of a bigger picture. So the amino acid sequence can then lend so squiggles, kinks, and even straight lines in an overall bigger picture. This would be a secondary structure. Tertiary structure is a 3D shape. So this I illustrated through a squiggle here. This would be this kind of on an even larger picture, so zooming out even more. And then we have the quaternary structure, which is illustrated by multiple tertiary proteins together. This would be maybe, like muscles, larger macromolecules, etc. Okay. Something I want to touch on real quick while we're here. So if you have ever cooked meat of any sort, what you are effectively doing is denaturizing the protein in the meats. What does this mean? So this means that you're taking that quaternary structure of a complete protein, complete healthy protein, and you're using either increase in temperature, increase in pH, or vice versa, decrease in pH or decrease in temperature, like you might see in frostbite, which then denatures the protein, so it causes it to unfold. And as we know in biology, structure really determines function, so after you have an unfolded protein, it is not able to effectively function in the capacity for which it's intended, which is important for us to talk about. Okay, real quick, since we are currently talking about proteins, I also want to mention something known as ligands. So ligands are molecules that can bind directly to proteins without modification. So an example of this would be oxygen in hemoglobin, so found in red blood cells. That's how we effectively transport oxygen to all of our surrounding organs. Also, proteins can function as enzymes. These are any protein that speeds up reactions. So they can do this by building molecules. These are anabolic reactions, or by reducing molecules, so breaking them apart. This would be catabolic. Essentially, proteins really have a lot of vital roles within our cells. Pretty important to know. Okay, so now we're going to go back to nucleic acids. Nucleic acids, like I mentioned, they have nucleotides. That's their monomer. This is what they would look like, the monomers, that is. So we have phosphate bound to sugar on a backbone here, and then a nitrogenous base extending off the side. There are 4 different nitrogenous bases found in DNA, so deoxyribonucleic acid. That is our you know, like the blueprint for our cells. That's what's telling our body what to make, what proteins, and therefore determines what is going on in all the cells. We have adenine, thymine, guanine, and citosine. And as you can see here, there is a relationship between each one. So adenine will conversely only bond to thymine, guanine will only converse to cytosine. I put U here off to the side because in RNA, that is the method by which we are able to get the message of the DNA outside of the nucleus. RNA only uses uracil for in place of thymine. So it will only bond to adenosine as well. So something really important to know about DNA is it's a double helix. So there are 2 strands of the phosphate and sugar backbone with these nitrogenous base projections that then pair up accordingly to the sequence. The sequence of the nitrogenous bases are held together in a weak hydrogen bond, and that's what forms the double helix shape. Let's see. Let's see what else we have to talk about. Hold on. Bear with me here. I guess the other important thing to say here is in nucleic acid, sugar is present. In deoxyribonucleic acid or DNA, that is a different sugar than what's in RNA. That and then also the uracil and the single versus double strand are the main differentiation between the two. Also, nucleic acids participate in energy production. So carbohydrates are broken down into what's called ATP or adenosine triphosphate. Adenosine being one of the nucleic acids. This is the primary source of energy in most all cells. Also, DNA is directional, so rather than illustrate it now, you do just need to know that they run in opposite directions of each other. So one head of the tail you can tell is the SOCO top because of the location of the carbon in the hydroxyl group that's bound to it. Vice versa, you can see which end is the end, so to speak, also related to that. All right, at this point in time, I think that's all I have for Unit 3. I'm going to take a little break here and I will be back with Unit 4.